Now, how does this round go? In front of every participant, there is a bulb, and you're going to cover it up with a fluorescent, orange fluorescent shade, and it'll all be lighted up. And you have four options: A, B, and C, D. Whichever they think is the correct option, they'll have to place that particular card, A, B, or C, or D there. And at the end of the time, we will see how many of them got the answers right. And all of them who answered correctly will all get 10 points. Right? This is it's 1, 2, 3, 4. So you'll all get it right, and then you get 10 points. Can we have a trial round, please? The first question. This is a trial question. No marks for anybody for guessing it right. Which of the following options is common to NCRM, Honda Motors Japan, Cathay Pacific Hong Kong, first steam engine powered and controlled airship? 1, they have a Japanese technological component. 2, all of them have transportation of cells or commodities as one of the research products. 3, all of them commenced on 24th September. 4th, all of them started without the presence or approval of US. Okay, place the numbers, please. Okay, and switch the lights on. So you have 1, 4, 4, 4, 4, and 4. Well, audience? Okay, the correct answer is 3. So this is going to be as silly as this, but then you'll have a lot of information as well. So this is a round where you can expect to score a lot of marks, so just be on the lookout. Okay, now we go for the real rounds. Do you all know what ostrix is? Is it a stem cell derived from ostrich? Is it an ingredient mixture that is used to culture stem cells taken after the comic character Asterix? Is it a transcription factor that is needed for osteoblast differentiation, bone formation? Or is it anagram for OCT2 telomerase 9 sequence? You have 10 seconds. Oh, you've got a lot of 3s and 1 4. None of you read Asterix, is it? We used to read it as a kid. What comics do you read? You read Asterix. Tintin. So you've never heard of Ostrix and Asterix? Definitely? No? Sure? It's not stem cells from ostrich, you sure? Yes, all except Team D have got it right. It is indeed a transcription factor that is necessary for osteoblast differentiation and is implicated in osteosarcoma formation. So, except for Team D, every other team gets 10 points. Okay, we have the next question. What is CMM, cellular memory modules? Is it a cluster of stem cells that are found in the hippocampus? Ability of cells to remember and propagate the gene throughout the entire development? Are the modules present in the Golgi apparatus of the memory T cells that are responsible for antigenicity? Is there any coherence between Alzheimer's and amnesia to mitochondrial inheritance? What does CMM actually mean? Okay, you have a lot of 2s, some 3s, and 1. You'll have to also give us some explanation. Why do you think it is 1? Huh? Guess? Yeah. Guess? Yes. Can I guess why you guessed so? Is it because the hippocampus is responsible for short-term memory? Is it because of that?
I can't guess why I guess so.
That's great. Okay, question 1 on 3. Why do you think it's 3? Again, it's a guess.
Memory, T cells, memory.
Okay, because the word memory comes there, modules also comes there. So too much of choices there. You have modules, you have memory, all of them there. So other questions don't have them, choices don't have them. That's why you've gone for 2, right? 3, that's why you've gone for 3. Okay, and 2? Okay, he thinks, why don't you speak it out?
I thought it was a reasonable guess amongst the other options.
Okay, he thinks it's the most sensible option among the 3. Okay, whatever others think. Audience, do you have two? So is two the flavor of the entire crowd here? Is that the answer? Okay, except for team B and team E, rest of the team gets point. The answer is indeed 2. We all have syllabus and we have modules. All these modules are shortened information. When you have cells that are grown, they have certain information that is encoded to be used at different points of time in their development. At some point of time, some of these are switched on. At some other point, some of the others are switched on so as to enable a cell to grow. This is called a cellular memory module, and team A, C, D, and F were all right. Okay, we go to the third question. What is ADAR? We all know of the Aadhaar card. Before the Aadhaar card, is it a temporary card that is given? Is it a gene that is encoding for adenose deaminase, and that is why it is called Aadhaar? Is it because the 12th month in the Hebrew calendar is called Adar, and that's the time when the stem cell potency is the highest? Or is it simply an anagram for alanine, aspartic acid, alanine and arginine, useful in cellular adhesion? Why is it 3?
Different.
What's your mother tongue? Tamil. Can you tell me all the 12 months in Tamil? Come on, guess. Anyone can guess the 12 months in Tamil? Okay. Thai. Thai Panguni. Margari, Chitrai, okay. That's 4. You have 8 more to go. Chitrai, Vaigasi. Okay, so we have so much of trouble knowing the Tamil months, and he knows all the 12 months in Hebrew calendar. That's good. That's true. Adar is the 12th month in Hebrew calendar. That's true. But then no one has found out that symbol potency is highest in that. That's not. So that's not the right answer. And 4. Any reasons for giving 4?
Like the other 3 were not looking right.
Okay. So anything that is in brown looks well for you. Okay. You can guess. Okay. And why do you think you chose 2? Reasonable guess. 2? Guess. 2? Guess.
No, we kept 3.
Okay. Team C, D, and F get 10 points. It is indeed the name given for a gene that encodes for adenosine deaminase, and since that is double-stranded RNA, it is called ADAR, ADAR. Okay. We go to question 3.4. Why are we speaking about HUINPCs? Is it an anagram for Hong Kong, US, India, Netherlands, and Poland who have come to form the cooperation for a rare gene sequencing? Is it after the Huizong factor, which is in the nuclear pore complex? Is it because the umbilical cord cell has been used for the treatment of nasopharyngeal carcinoma? Or is it because epithelial cells in the human urine have been transformed to neural progenitor cells? Because all of you are guessing, let me give you some hints. Who was the first non-Congress Prime Minister of India? Moraji Desai? Okay, think of Moraji Desai. Some things are associated with him. Some idiosyncrasies. He was a good Prime Minister, I think, well before my time. But some idiosyncrasies are associated with him. If you know what idiosyncrasies are associated with him, probably you'll get this answer right. My God. So because you're all getting good guesses, I've given you a good clue. So who gets the points? I think I should get the points, right? So that's indeed the right answer. Epithelial cells in human urine transform to neural progenitor cells. So that's the right answer. Question 3.5. What's genetic erosion? Some serious stuff. Is it a frequency of change in a gene of a population due to random sampling? Is it loss of particular genes because of the inability of animals with that gene to meet and breed? Is it the erosion of a population because they inherit more and more of the father's gene and less of the mother's gene? Is it mutations in the gene that erode the original phenotype of the organism? We'll give you some 20 seconds to think on this because it looks like a difficult one. You'll have to read through and make up your mind. You can use your calculators if you have one. This is interesting. When we talk of erosion, there's always erosion that happens when two things meet. Talk about sea erosion. When sea meets the shore, sand is eroded. When you talk about erosion due to wind, when wind meets a rock, the rock is eroded. Talk about cultural erosion. When American culture interacts with Indian culture is eroded. Talk about culinary erosion. When different cuisines meet, there is erosion. Is it not? And this is an erosion, maybe the only erosion that happens because two things don't meet. And the answer is, when animals that carry a particular gene do not get a chance to meet and breed, that particular gene is not passed on to the next generation, and therefore you have genetic erosion. And we have team B, team E, and team F giving us the right answer. Is this the explanation you thought? And E, he thought about exactly the same, and that's the reason why he's got this answer right. Okay, we'll give these teams full marks. And the last question in the blah blah. Oh my God, wait. Okay, we all know this. This is Petri dish. Does it exist like this before? What is this? This is a plastic Petri dish. Before you had Petri dish made glass. Fine. Okay. Why is a Petri dish so called? It was named after the German bacteriologist Julius Richard Petri because it is a polyethylene tetramer recyclable impervious dish. Petri means growth in Italian, so anything that helps it grow is Petri. Petri is an island near Bahamas where this was first developed. Number there. Audience? Okay. The answer is indeed 1. Except for team D, everyone gets answers. You're so much involved in Italian, is it? Okay. Now we have the audience questions. What is, among this, what do you think is gap? It's a very straight question. Don't have any nasty thoughts. What's gap? What's gap among these?
Sir, Great Ape Project.
Yeah. Yes. A lot of non-human primates were taken up for studies, especially because if anything that is successfully replicated in them becomes very useful to take forward to human trials, and therefore there was a lot of ill treatment that was happening to these primates, and therefore a movement came which is known as the Great Ape Project, which is an international movement that wants to defend against the abuse of these non-human primates. Fine. One more audience question. What is junk DNA? DNA inherited from the father. Father's junk anyway, so DNA inherited from the father. DNA material accumulated during viral infection in human cells. The portion of the DNA that doesn't get transmitted during cell division. All portions of the DNA which have no human function. 4. Okay, which is the answer? 4. Which is the answer? 4. Who gets the pens then? Everybody. Okay. So we don't have enough pens, so we'll stop here. At the end of this round, the scores are, wow. Suddenly the scores have all become very impressive, haven't they? We promised that this round is also all making you going to look great. Well, at the end of this round, we have Team C, Madras Medical College, right on top, 70, followed by MIRM at 65. AC Tech is 60. Anna University and JIPMER are 4th and 5th. Chengalpet, 30. Just 20 more points or 30 more points behind at least the second place team. Catch up.
So from blah blah we come to terra firma. So there's one more blah blah, okay. That's the good news for you. There's one more blah blah. Don't get disheartened. This blah blah is when you make up, and all these tough concepts come in the blah blah. It's sugarcoating the tough concepts, so don't think it's a joke. We now start with the fourth round. Starts with one. Team A, right. Ready? We are talking about those intestinal stem cells. It has been puzzling us for a long time. We have those cells in the crypts known as columnar CB cells and the +4 cells. And most important is that we are stuck up with those markers. Whenever you read about those markers, suddenly one guy comes up with a study and say they are markers at the embryonic level, and these markers change when they become adult cells. So these markers keep changing and changing. But somehow, after a long study, they have come to various standard markers for intestinal stem cells. You know that the intestine, it regenerates every 3 to 4 days. So I'm giving you a few stem cell markers for the intestinal stem cells. One is BMI1, the other one is TERT, the other one is HOPX, LRIG, and LGR5. Of these markers, LGR5 is what keeps hitting you day in and day out. It has been accepted as the marker. Simple question, quiz question. Expand LGR5. Don't tell me it is an intestinal cell marker. I want you to know what is LGR5. It's not Lucy and grandma and all that. LGR5, it is an intestinal stem cell marker. Markers are very important in stem cell morphology. Without knowing markers, you cannot study stem cell behavior and morphology and reprogramming. You're ringing a bell for me or for them? Oh. She doesn't like me talking. Hey, no problem.
Labercon growth receptor 5.
Good try. You're from which college? JIPMER, yeah. Really good try. You're in the molecular biology department, huh? You're nearby. The hostel is nearby? Or you cross the department when you go for classes? Whatever, he's been influenced by molecular biology. Team B. It's a very easy question, huh?
I think GR stands for glutamate receptor.
Yeah. You think means I also may think. But you seem to be thinking sometimes on the right track. But in the blah blah, they were thinking wrong, huh? You get marks only if you know the entire thing, okay. Otherwise, it's not fair. See, you can use your chromosome computers, chromosome calculators.
Large intestine growth receptor.
Good try. I never thought yellow is for large intestine. No, good. I think we can write to them. Team D? Is it? I want the full answer. Team E?
Leucine-rich granulocyte receptor. Fine.
Team F? Okay. It was a question for A. Audience? It's leucine-rich repeat containing G protein receptor 5. I'm sorry, I wanted the full answer. Sorry, we couldn't help you out. Okay, this is a tough question. You know this, what is known as the Eureka moment, right? Eureka moment. No, you're having a bath and you're having your exams and your cell phone goes off, and suddenly the equation balances, and you feel like running out of your bath and saying, I have discovered it. A lot of these mathematical equations and physical formulations are conjured up in our dreams. These molecular structures sometimes appear like monkeys and they digitate. That's how major molecular structures have come around. This is regarding a stem cell. This was discovered by Gregorio Chaisenbach at the UCLA lab. This team stumbled upon a group of stem cells post liposuction. The patient had undergone liposuction, and suddenly the current and generator failed. The cells were submitted to extreme hypoxia and physical stress. But what survived after this hypoxia and physical stress was a group of stem cells which were seen post liposuction, published in the year June 2013. These cells were earlier discovered in the Tohoku University among bone marrow cells. What is the name of this accidental stem cell? I want the abbreviation and the full name. An accidental stem cell. So next time, in Tamil Nadu, I think these power cuts are quite frequent, no? So each time there's a power cut, you go and check up whether there are any new stem cells. In CRM, you have 24-hour backup lab, no? I think you should not have that. That's why you're not picking up the new cells.
Okay.
Okay, team C.
Eureka cell.
It's a good try. No, unless you know it, it's difficult to. Or you should have been in the lab.
Serendipitous.
Serendipity, okay. Which country was known as Serendip?
Sri Lanka.
Sri Lanka, good. What is serendipity? Searching for a needle in a haystack and finding the needle. That's serendipity, right? Team D. Tough question, no? Because even I have not seen this cell. Team F. Team A.
Adipocyte-induced pluripotency.
Audience, I think you used your cell phones and got the answer, right? There are no mobile jammers. These cells are known as Muse cells, multilineage stress-enduring stem cells. So this is a new accidental stem cell. So in Tamil Nadu, the possibilities are endless, right? Question to team C. If you read those papers, a lot of papers are done by Chinese and Indians, right? Most of the papers done in the United States are by Indians and Chinese. I think the Chinese do seem to dominate. This is a publication from Anura Rambukana, a regeneration biologist at the University of Edinburgh. Those Dolly people, okay. They made a startling presentation in the January 2013 issue of Cell about a bacteria turning adult cells into stem cells. This bacteria is very relevant to India. So this bacteria turns adult cells into stem cells. What is the bacteria disease and what is the cell of target? Question to team C. Quite relevant to India. Helen, time. Oh, you have become relaxed, no? This is also timer, no? Okay. Pause.
It is Mycobacterium tuberculosis.
Mycobacterium tuberculosis. And what is the target cell? It has to attack a cell, no? Mycobacterium attacks so many cells, no?
Macrophages.
Macrophages. Okay, we'll keep it in the back burner. Team D. D, use your calculator.
Is it dendritic cells?
You're asking me a question. I'm the quiz
Dendritic
master.
cells.
I'm the quiz master.
Dendritic cell.
Huh?
Dendritic cell.
Dendritic cells. Okay, what is the bacteria? Dendritic is not a bacteria, right? They are in
Going
basically
with Mycobacterium tuberculosis.
Yeah.
MTB and the
Mycobacterium tuberculosis,
Tuberculosis and
and
the-
dendritic cells. So you're trying to say it's not macrophages. They have a preference for dendritic cells. Why? Some markers, signaling, trafficking, niche. Okay, we'll keep it in the back burner. So be waiting. Okay, team E.
Mycobacterium tuberculosis and lung cells. No?
Yeah, lung has got so many cells, no? What cells? Tracheobronchial, bronchiotracheal, laryngotracheal.
Bronchial.
Bronchial, sure.
Yes.
Not laryngotracheobronchial.
Bronchial.
One diagnosis which I never understood in medical college was laryngotracheobronchitis. I never knew where that organism was. If you didn't know to make a diagnosis, you say the patient has got laryngotracheobronchitis. Are you still using those diagnoses in college nowadays? No. Very smart, huh?
Mycobacterium leprae and its Schwann cells.
Microbacterium leprae and Schwann cells is the right answer. Give them a hand. So what happens? What happens?
Sir, I think my classmate can tell. He presented the paper.
Oh, you done a paper, huh? So you're working with, oh, you're Anura Rambukana. I always have a feeling that I'll end up with one of these papers. Huh? Okay. So what happens? Mycobacteria attacks the leprae, attacks the Schwann cells. So Schwann cells loses its function and the nerve is exposed. Okay. So what all can come out of this? So you can use bacteria to reprogram adult cells into stem cells and kill the bugger, right? So that's how the study goes. So they say we feel that in future we can use bacteria, convert adult stem cells into stem cells, kill the bacteria, and we can do away with all those Yamanaka factors. Basically, all those papers published post Yamanaka, they want to do away with the Yamanaka factors. I don't know what wrong Yamanaka has done. And I think even Yamanaka may come up with a similar study, no? The end of Yamanaka factors. So question to team D. So Kalpana Sharma, no? Sorry. Chawla. She's so much in the news that it doesn't hit you. Okay, she's without the space shuttle and all, no? If I'm not wrong. Okay. So this space shuttle has been going up and coming down, right? Sometimes it doesn't come down. Sometimes it doesn't go up also, right? Question to team D, yeah? Tough question, huh? You're interested in NASA? Aeronautics? No. Actually, if we had opened up the NASA website 2 days back, it was not opening because the American economy had shut down. All these NIH and PDB websites are not opening. I think we had a head start. If someone missed it. They have more shutdowns. Okay, let's not dilly-dally. What was the special payload about Space Shuttle STS-87 in the year November 1997? The Space Shuttle STS-87 had a special payload when it went up to space. What was the special payload? And what was the insignia on the Space Shuttle? What was the insignia on the Space Shuttle? You don't like flying, huh? You're phobic. You don't even seem to like the question at all. Team E, I want perfect answers, not just so answers.
Cancer cells were sent to space and they organized themselves in 3D and they were more resistant to tumor.
What cancer cells?
Breast cancer cells.
Breast cancer, okay. Dolly, okay. Team F. You're also not interested in space. Huh? I think there was a big discussion on space. Anyway, I'm happy that nobody came for that discussion here. Team A can do it.
Bacteria were sent and
What bacteria? Salmonella or Staphylococcus or Pneumococcus or I forgot. I mean, that's all I can remember. Lepra bacilli also, Mycotubarium.
salmonella.
Salmonella, yeah, the food poisoning thing. Huh? So they wanted to see whether the astronauts got food poisoned or not. Okay, good try. Team B.
What? 2 spiders were sent on the payload.
2 spiders. Huh? Spider-Man. No.
Spiders.
Okay, male spider or female spider?
2 were females.
Good try, man. This is very interesting. Point. Team C.
It's a dog
Dog.
named Laika.
Dog and all the old days, no? These Russians sent it, no, to Sputnik and
Monkey.
then the monkeys went and all that. Okay. It was a question to team D. Audience. Tough question. Now you made an easy question tough. Audience? Okay. That entire shuttle was known as a microgravity shuttle. So the entire experiments were done on microgravity. The insignia on the shuttle was mu g. All the experiments in the shuttle were on microgravity. And if you have any doubts on microgravity, we have a big pista here, Dr. DDBR. He is our in-house specialist on microgravity. In fact, it was he who introduced the concept, I think 2 years back, no? So every prelims there's one question on microgravity, am I right? He really loves it, but there's something known as hypergravity also, no? So I think next time we'll go into hypergravity. Okay, G is equal to 4 pi r squared, no? That's the only thing I remember from. Answer is it's mu g, microgravity payload. Yeah, that's the microgravity experimental module. Okay. Somehow I've had a big fascination for viruses. It's because we see these lot of conjunctivitis and these things don't get they don't get well, no? They keep going from one eye doctor to the other eye doctor. And each patient who comes thinks that we have God and we can clear it off. It doesn't clear. So I started reading a lot about viruses so that I can defend myself with the patients. But still it's no end game. Okay, question to team E. This virus, there are very nice viruses. Okay. Not all viruses are bad. Some viruses are friendly fellows. They keep coming into your body and going out. This virus integrates into the host cell at a specific site on chromosome 19. Okay. This integrates at a very particular site and always keeps integrating into that site. 19q13.3. Just keep checking it up. This is a very innocuous virus and is now used as a viral vector in more than 80 clinical trials. Name the virus.
Retrovirus.
Retrovirus. You want the name, specific name, not the family. Don't tell me it's those Picarna, all those viruses. Tell me the specific name.
Rao sarcovirus.
F. A.
Adeno-associated virus.
Adeno-associated virus is right. So what is great about this AAV virus? It specifically integrates only at the chromosome 19. So you can integrate genes. And this is known as a genomic safe harbor, right? Good. So there's a big viral gang. So things in Facebook also can go viral. Okay. Last question. So like animals, plants also have stem cells, right? Obviously, you know. So Plant stem cells are characterized by the location in specialized structures. What are they known as?
Shoot and root apical meristems.
Shoot and root apical meristem is right. So DDPA, we were always wondering which stem cells this Manipal team was into. Now we understand. A lot of work going on plant stem cells. In fact, I feel that those of us into stem cells can initially take up work on.
Sir, actually, I recently read through it CLV3 WUS pathway that is activated in
Yeah.
shoot and root apical meristem.
Yeah, I don't know all the pathways, but the
that is-
pathway that you know, I don't know. Yeah. Actually, like Dr. DDPA, I love those pathways. But you know, it's like falling in love with a beautiful girl. Every time it's a new pathway. As an ophthalmologist, they say, no, this color vision is like a beautiful girl. You never follow the same girl because every day there's some theory about the color vision. So nobody's bothered really about color vision. Yeah. Next, blah, blah. Okay. Can I have the scores? So looks like Manipal is because of. So you're doing work on plant stem cells, huh? But I think, I mean, it's eco-friendly, no? I mean, you're not working with monkeys and. Sincerely, I thought you could do something. Yeah. Audience, this nanog is known as the master gene, no? So what is a nanog? Why is it called nanog?
The land of ever young. The land of ever young.
So this everybody knows, huh? This is the question we asked in the first, last year we all asked, huh?
Sir, because
You told me you asked only in the first year.
I can tell you the controversy.
Visual, okay. This is always a fallback question, huh? Good. Can I go for the visuals? Visuals? Scores சொல்றீங்களா? So after this, there is blah blah. So which means there is still light at the end of the tunnel. The big light at the end of a long tunnel. Okay. Okay. This is okay. Maybe it's a visual from Alice in Wonderland or Alice in Blunderland, whatever. As you can see, two living animals join together surgically and develop a simple shared circulatory system. Usually it is heterochronic, older rabbit or mice with a younger one. But the first study was done in the year 1864. It is found when this kind of surgical joining, or what we see as Siamese twins in our medical literature, the older rats or rabbits, when thus joined, they live 20% more longer, and it leads to a rejuvenation of aged progenitor cells. What we don't know is whether the younger rat also becomes older, right? The older rat becomes younger, but we don't know whether the younger one becomes older. It's because of those growth factors and all that blah blah. Okay, the question is, what is this procedure called? Question 2? F. You didn't read any article on this? But I'm happy you read on plants. Huh? You're a very eco-friendly guy. So what is this procedure called? It's not done in human beings. You can't have grandmother with a grandson. I mean, maybe. Team E? Team E. I think you can try it in your labs, no? Of course, you can have an older plant with a younger plant. Huh? No. That's grafting. D. A kind of grafting. C. Yeah, it's a specific word. You cannot tell share and care and all that stuff. No. Tempted to say share and care and share. B. A.
Shared aging.
Very cynical guy. Huh? Audience, you didn't do your internet search and all, no? It's called parabiosis. So you can try it in your animal labs, provided the ethical questions are sorted out. But then you have to go to Italy. So there is a famous ophthalmologist. Anybody from ophthalmology here? Of course. You all been to an eye doctor, right? Most of them were wearing glasses, right? A famous ophthalmologist by name Payman who comes to Bombay. And he has discovered a new technique of suturing. And the audience, I mean, very learned and distinguished doctors in the audience, top doyens of India. They ask him, how do you suture? And what he does is he takes his shoe and keeps it on the podium and shows them how to suture. So whenever you see a shoe, I always think about that ophthalmologist. But of course, our Indians didn't like him. Okay, this has nothing to do with that. That's a shoe and that's a shoelace, right? No doubt about that. That's a chromosome and that's a telomere. The difference between chromosomes in plasmids and virus and us is because in virus they are circular. In plasmids the chromosomes are circular. In humans and other animals the chromosomes are linear. So you need something at the end of the chromosome to stop the leakage of genetic material and also to stop those linear chromosomes from joining each other. I'm trying to make it tough. It's a question for team E. Now let's come back to the shoelace. Okay, that's a telomere structure in the shoelace. Technically, what is that structure called in a shoelace, which represents the telomere in a chromosome? In a shoelace, what would you call it? You know, in our young age, we had 1, 2, buckle my shoe, and what was that? 3, 4? Shut the door, huh? 5, 6, pick up the stick. 7, 8? 9, 10? Yeah. Okay, but the answer is not in that. I hope the answer wasn't that. I thought I was... Okay, what is that structure called?
Loop sequence.
No. You don't... Okay. D. You think like you're going to Bata's showroom and in your lace that is lost, you don't tell him, I lost that in my lace. You ask for something specific. Of course, we don't do it because we buy a new shoelace. D. This is very important, huh? The Nobel Prize in 2009 for medicine went to people who chanced upon these telomeres and defined the telomeres. So telomeres are medicine, and we know that Dolly died because of telomere shortening. Team C?
It's a lace cap, a lace protein.
Lace cap is good. Team B?
Telum.
T, okay. Team A?
Lace link.
Lace. F? Audience. It's called an aglet, okay? Aglet, A-G-L-E-T. So next time something happens to your aglet, go to the Bata shoe and say, I want a new pair of aglets, because they are not going into the eyelets. Those holes in the shoe are known as eyelets. But, where are we? Yeah. There's one more round, no? Blah, blah. Yeah. So we talked about those green fluorescent proteins and red fluorescent proteins long time back. Next slide. If you go through those NIH websites and this protein database And Swiss Prot, you see the structures. And if you click in the bottom, this thing starts rotating, and it rotates this way and that way, alpha, beta. It's so interesting, but you can't really understand it. Huh? I've burnt myself doing all this, but still back to ground zero. We have things like in vivo. Question to team? D. In vivo, in vitro, in situ, in utero. What is this procedure which is performed on a computer or via computer simulation? What is it called? It was presented by Pedro Miramontes, a mathematician from Spain in the year 1989. In vivo, in vitro, in situ. What is this called, which is performed on a computer or via computer simulation? There are a lot of softwares, nearly about 40 softwares, how to get up those structures. What is it called? I heard for biotechnology students, this is the first chapter in any book. So, it's a question too? Yeah, it's over. See. சும்மா சொல்லுங்க.
Inteco.
In?
Teco.
Inteco looks like some festival in IIT Chennai. Yeah? That's Mardi Gras or Indigo. Indigo. Okay, team B.
In silico.
In silico is right. Give them a hand. Huh? I guess a lot of people were not aware of in silico. Am I right? Yeah. So you go and check up in silico. A lot of softwares, and as usual in those early days, most of these softwares are free. And then when you get hooked on it, they become payable. Okay. Question to team? Okay, that is an equation by a famous statistician. And he worked on cancer and smoking, right? We all think that cancer and smoking is just like that, but it has taken nearly 15 years to prove the link. Anybody doing statistics, epidemiology? Nobody, yeah? It's not mandatory. There are a lot of things known as cofactors, right? You just cannot say, I think, Saravanan is done in London, right? Okay. So I better keep my mouth shut. Okay. So this equation, basically what this team has found is that the incidence of cancer in humans is much higher than the incidence of cancer in whales. Although, if you use common sense, whales will have, supposed to have more cancer or more mutagenesis than humans, because they are bigger structures, more cells, more lineages, more differentiation, more transdifferentiation and all that stuff. This is a paradox. What is this paradox known as? So the equation says P is the probability of getting cancer, mu is mutations, D is again divisions, K is mutation pathways, N is the number of cells, and M is the number of cells. This is all chumma blah blah equation, but it is true. Okay. But only Saravana Ganesh can understand that. What is this paradox? When an animal much, much bigger than human beings is prone to less mutagenic events, and man, after all, he is such a small guy. I think he fits into the mouth, right? Jonah's whale. What is this paradox known as? Question to team C.
Hayflick limit and dolphin paradox.
Huh?
Dolphin paradox and Hayflick's limit.
Okay. B.
It's called Peto's paradox.
Oh. These guys are great. It's Peto's paradox. So Peto was an epidemiologist, and he was involved in meta-analysis studies along with the Bradford Hill and smoking cancer team. Last two questions, huh? Oh my. I think the easiest question, B, it's a sitter. Huh? Can you go back? No, this is the first one. No? Back. So this is a landscape. Huh? And you know this Palani Hills and Ooty Hills and Mudumalai Hills and all. So they had to make a topographic map. You have a peak and you have descent and you have ridges and you have the end stages, right? So this is a landscape. It's a map. So you can see all cells, they follow different routes to reach their maturity. And basically once a cell becomes mature, it cannot go back. The cells can only come down. Next one. And of course, the great induced pluripotent cell transformation, anything can happen to any cell. Of course, Yamanaka, he had a tough time in taking up the cell back. Yamanaka said, we'll transform, but we'll take the cell up. But there is a new concept known as lineage reprogramming. I think, that's my gut instinct, lineage programming is the in thing. Manipal, are you with me in this? Yeah. Okay, that's mean you're not doing it. Okay, fine. It's conversion of mature terminally differentiated cell type into another mature type. It's mature to mature without going through de-differentiation. So that's all blah blah. Okay. Now what is this landscape known as?
Hillis plot.
Huh?
Hillis plot.
Spell it.
Hillis plot.
Hillis block.
Plot.
Landscape. A.
Trans
This was published in the year 1957.
Transdifferentiation.
Yeah, differentiation, all that stuff, totipotent, pluripotent. This is the latest interpretation of the landscape. Sam is very excited about this question, I don't know why. Team F, when you're silent, they're silent, huh? So there is some cue going on, okay. But this is a sitter, huh? I mean, this should be framed in your rooms. I think 4 or 5 years back, we were trying to create a model of this landscape. Team E?
French flag analogy.
Huh?
French flag analogy.
French flag, huh?
Analogy.
Yeah, you're confusing me, man. D? As far as I know, it's not French flag. But that is all that we have to know now. D. Okay. Have a look. These are ridges, and you get lineage programming from one mature cell to another mature cell. C was
It is Conrad Hall Waddington.
Conrad Wallington epigenetic landscape. Right. So even though they knew the answer, they kept as cool as a cat. Huh? I thought they didn't know and they'll come out to something horrible. Okay, last question. A, huh? Okay, this is the people who visualized or dreamt about the Higgs boson. Okay. Francois Engert and Peter W. Higgs. So we are not asking you about what is Higgs boson, and I think it is the stem cell of space. Huh? Yeah, something like that. Okay, let's think like that. When they were announced the award, Peter Higgs came out with an interesting statement. He said, this Nobel Prize is going to impact a kind of research. It's basic research, but he used a beautiful word, and I think it's the first time somebody has used this word. This research means it's research without return of investment. Basically, when you submit a research proposal, you make a 2-page statement at the end. Because of this, we are going to reduce this, and the economy of India is going to grow. We had a discussion. There are a lot of people who can write such papers, you know. Any paper, they'll make it's called as a justification. This is research without justification. This is research without looking at the economic impact. This is research without a goal. This is just curiosity-driven research. I'm curious, I research. What is the word that Peter W. Higgs used? In Europe, there are institutions who fund this kind of research. So it's not a dull and drab question, it's a question to team A. It was in all the papers. It was in all the Indian papers, all the internet papers. A very specific word he used. In fact, but for that, I wouldn't have known this word. A. You have a blah blah round to make up, so don't really worry. That's why you get the blah blah there. F. E. Why you become so silent on this question? Man? You're thinking about this boson and all, no? D. C.
Intuitive research.
Huh?
Intuitive research.
It's a kind of intuitive research. It's basic research. But that's a lovely word for that. B.
Utopian research.
Utopia is also wonderful. Audience. It's called Blue Skies Research. There are agencies known as Blue Sky Foundation in Europe. So if your proposal is not accepted by the government, you look for a blue sky. So Preeti says the time is over. Back to blah blah. Yeah. Thank you. We have the last round, and Dr. Saravana Ganesh, who was my junior, am I right? Yeah. I was a fine arts secretary, he was a fine arts secretary, and Sam was a fine arts secretary. And he's a cardiac transplant surgeon. He's got, if I'm not wrong, 3 FRCS degrees from London. And he's so modest, he says he's got only 1.
Thanks, Johnny. Right. I think Sam's sitting on my head saying there's only 9 minutes left for about 6 questions. So let's get on with business. Right. First question. What are tanycytes? They are T cell aggregates in niche in cell media. First stem cells processed from Tanya. Anybody got a girlfriend called Tanya? Progenitor ependymal cells in the brain that help in neoneuronization. And the cells discovered recently that help in the tanning of skin. Your answers on the desk now, please. You're going to switch the lights on only after you place your cards in, so that you don't copy from others. Right? Cards in, everybody? Lights on? Wow, that's a full house. Very good. Give them a big hand. Everybody's got it right. Okay, next question. What or where is a Dewar layer found? I think everybody's smiling. I think it's a sitter. Like Johnny says, it's a real sitter. Okay? In the brain, in the Golgi apparatus, in the eye, in the hypothalamus. Answers now? Okay, lights on. Oh, wow, that's a surprise. What's your number there? So, what do you think it does in the Golgi apparatus? It was a guess, okay. Well, I didn't expect it from MIRM. They think it's in the brain. Why do you think it's in the brain, guys? Okay. For guys who've got the answers right, it's number 3. Who was Dua?
Indian, Ahmad Dua.
Well, you can't call him an Indian. He's a British guy, but he's an Indian origin guy. His name is Harminder Singh Dua. He's an ophthalmologist who actually found a new layer in the cornea. You know, we were traditionally taught there's only 5 layers. I think he found the 6th layer recently. So answers goes to A, C, D, and E. Okay, good. Next question. What is a degron? I think that's the new degree that Dr. Sam's team is offering from this year. Or is it the degradation of neurons after an injury? Maybe it's a unit of measurement for stem cell lines, I think, you know. Or it could be a specific sequence of amino acids in a protein which, you know, directs and says where the degradation should start. Answers on the desk now. Okay, lights on. Whoa, what do the audience think? 3, okay. People have thought 4. Why did you think it was 4? Did you read it anywhere from any journal or? No? Guess? Right. I thought somebody would have gone for 1. It would have been really good fun. Said that. Anyway, yes, 3 is the right answer. It is a specific sequence. So, you've got full marks for E and F. Okay, next question. Homeo box. Oh, there's a big shh. I know. This round was meant to be easy, fun. Not for those guys, but for you guys. But it was also supposed to boost up the scores, you know. So, as you know, all these questions for each round was designed by each quiz master. So you can see how dumb I have been. You know, I've been choosing all very easy questions. So that's how I stand. Anyway, it's a DNA sequence found within genes. It's a box to carry homeopathy medicines. It's a homeopathically generated stem cell line. Is there a stem cell line that's homeopathically generated? Does anybody know of anything like that? Are you sure that there's nothing like that? Okay. It's a stimulator for demonstrating the pathways of vascular endothelialization. Answers on the desk now, please. Lights on. Wow. Again, MARM comes a cropper, isn't it? You put 4 in there. I just saw you put 4 in there. You actually come a cropper. Why did you put a 4 in there, guys? No, I don't know. No, I saw you put a 4 in there. Yeah. What was the reason? Well, I don't know. They don't have an answer. I think they were trying to change the card or something. Anyway, 1 is the answer. So everybody except F gets the right answer. So it was not a dumb question after all because MARM gets it wrong. Okay. Right. Something close to my heart. You know, I'm a heart surgeon. It's very close to my heart. I like treatment for heart failure because I do surgeries for heart failure, which itself is a concept among heart surgeons. What is the STOP HF SDF1 trial? It's a trial to identify factors to prevent heart failure following heart attack. It's a novel gene therapy trial for ischemic heart failure. It's a landmark case in the US Supreme Court which paved the way for stem cells in ischemic heart failure. Safe to try, but otherwise poor treatment strategies in heart failure. Answers on the desk now, and please don't change your cards. Yeah, light on. Okay. Wow. Tell me, why do you think it's option 2? Chengalpet, you've been very quiet for the whole afternoon. It was a random guess, is it? Okay. What about you guys? 3. Sorry? Reasonable guess, okay. So there's an unreasonable guess as well, is it? Okay. 1, guess again. Audience? Well, that points to only one thing. Nobody reads any journals these days. Anyway, the answer is 2. So, you know, it's actually a gene therapy trial, and we have a quiz on gene genetics, and nobody actually has the answer. Fantastic. Right. Well, I think we still have time for the last question. Okay. Uniparental parthenoids. Lights off. They are stem cell lines derived from single parent offsprings. Unwanted byproducts of in vitro fertilization. A new way to create babies in humans? Possibly. Is there any other way? Cancer stem cells derived from a single parent used to treat their children. Answers on table now. Has everybody got their numbers on? Okay, time's up. Lights on. 1, 2, 1, 1. What's that number there? What's your number there? Okay. So, what do you mean by stem cell lines derived in single parent offsprings? You've put 1 on your option. Was it a guess again?
Just a guess.
It was a guess again. What do you think?
Something similar to parthenogenesis.
Sorry? So what are parthenodes? We know what uniparental is, but what are parthenodes?
Offsprings derived from a single cell without any fusion, without cell fusion, without fertilization.
Without fertilization. Okay, so the only clue there which says fertilization is number 2. So the answer is number 2. Okay, so B and E get full marks. Audience? One question? Well, Sam tells me there's one question, time available for just one question for the audience. So you guys can relax now. What is a ring? Oh, wow. There you go. She gets the prize. Okay. Fine. Thank you very much. Okay, now Dr. Didipia will announce the scores for us. Here we go.
AC Tech, after a very, very quiet beginning, you made it up in the end and narrowly come first in the 8th Physio Cup quiz here. Full marks to you guys. MIRM, you've always been 1 or 2 or there right about in these last 7 years. Well, this is your time to be 3 on the list. So you are 3rd. You were, I think, leading until the end of this, at the beginning of this round. So you with a full century, but yet 3rd. So the second team is the team from Madras Medical College who've been very consistent and probably missed out some easy questions. Congrats for being 2nd. And good show by the rest of the teams there. I think this is one of the highest scoring quiz rounds that we've ever had. We've never had the team that wins score more than 70 or 60 in the past few years. So I think grand ovation to all the participants here for scoring maximum questions right in this quiz.
So, and the team which prepared the quiz is approximately some 30 to 45 people have worked on this. And we had great quiz masters, Dr. Saravana Ganesh, Dr. Jani, and Dr. Didipia. May I request Right Reverend Father Francis Xavier, Director of LISET, to come in front and then kindly hand over a small token of appreciation to all 3 quiz masters and blah blah masters. Yes. So, thank you all, and we are 5 minutes ahead of schedule, and it's a very good thing. So let us all gather back at say 15:20 sharp after a quick